So basically in this video, we will discuss about the active transport and related things in bioinorganic chemistry. So what is the importance of electronic structure of metal center? As we know, the role of metal in biomolecule is of structural and catalytical role we have. The metal ions have structural as well as catalytical role. Sometimes a metal ion can do both the roles. Okay, so metals that have catalytical role will be present at the active center of this biomolecule, which is likely to be a metalloprotein. So reactivity of metalloprotein can be defined by the nature of metal and particularly its oxidation state and electron structure. Okay, so the role of electronic structure and spin state, it's very important. Electronic structure and spin state of metal center defines the chemical reactivity of spin redox center. Chemical reactivity as redox center. Okay, so redox center, as we know, it controls the efficiency of accepting or donating electrons. Also, electronic structure of metal center defines its chemical reactivity as Lewis acid. What is a Lewis acid? Electron pair acceptor, which enables it to bind to ligands for transport, activation, and reaction. Also, electronic structure and spin state of metal center permits its investigation and characterization through electronic Mosbaer and ESPR. Okay. Now coming to the problems to be considered. Basically, the first thing to remember is transport of ion. It is very important because all cells will be acquiring the molecules and ions that they need from their surrounding extracellular fluids. Okay, and this extracellular fluids, from this extracellular fluids, these ions or molecules will be moving in and out through its plasma membrane. So there are unceasing traffic of molecule. For example, glucose, sodium, and calcium, these may be going in and out through this plasma membrane. Also, in eukaryotes, there is a transport in and out of membrane-bounded intracellular compartments. That is, in and out this membrane bounded intracellular compartments. Also in eukaryotes, transportation of ions are seen. So it is not only seen between it is not only the cell will be acquiring molecules or ions from their extracellular fluids through its plasma membrane. Not only that, but also from this intracellular compartment. Between this boundary bonded membrane bounded compartments, intracellular compartments, ions are moving. So the problems under consideration basically relative concentration, because diffusion or osmosis, this is a phenomena where molecules can be transported. Okay, these molecules or ions will be going spontaneously down their concentration gradient, that is from a region of higher concentration to a region of low concentration, and the reverse is not possible. The reverse is not permitted by diffusion. Diffusion is simple. Diffusion is the basic way in which more ions or molecules are transported. So here molecules and ions can be moved against their concentration gradient by active transport. So in this video, we will see details on active transport. Okay, and this active transport will need an energy, and that is acquired through ATP. Usually, this energy expenditure is compensated by ATP molecules. This is the first problem under consideration while discussing the transport of ions. Secondly, the lipid bilayers are impermeable to most of the ions. Why this lipid bilayers are impermeable? Because this lipid bilayers, they will be they are permeable to water molecules. Also, some of the uncharged small molecules like oxygen and CO2, this lipid bilayers are permeable, but they are impermeable to most of the essential molecules. Hope it's clear. Okay, these are impermeable to most of the essential molecules and ions. Like these lipid bilayers are not permeable for ions like essential ions, potassium, calcium, sodium, fluorine ions. Also, small hydrophilic molecules like glucose. Also, for macromolecules like proteins and RNA, these lipid bilayers are not permeable. Also, um, okay, as we know, lipid bilayers are permeable. They allow the transport of in and out of water molecules and some uncharged molecules. They can diffuse freely in and out. Diffusion is the process. As you know, diffusion of water through a plasma membrane in and out of cell, it is nothing but osmosis, right? So now the task is to overcome these two problems. That is the relative concentrations. Like we should go against this relative concentration, or molecules or ions should be moved against their concentration gradient, and that process is known as active transport. So we should discuss that. Also, this impermeable lipid bilayers. So how to resolve the impermeability of this lipid bilayer? So mechanism by which cells solve the problem of transporting ions and small molecules across their membrane. First one is facilitated diffusion. So what is facilitated diffusion? Transmembrane protein created water-filled pore through which ions and some small hydrophilic molecules can pass by diffusion, and the channel can open or close according to the need. So facilitated diffusion, as the name suggests, facilitated diffusion. It is not a continuous diffusion process. It is facilitated, right? Transmembrane protein. Okay, transmembrane protein. These can create water-filled pore through which ions and small hydrophilic molecules can pass. Okay, through diffusion. So, and these channels created by this transmembrane protein or water-filled pores through which ions can pass by diffusion are of, like, they can open and close. Okay, according to the need. Actually, we will see this facilitated diffusion in detail next page. Then active transport. Active transport is nothing but, as we said, this transmembrane proteins, or we can call this transmembrane proteins as transporters. They use the energy of ATP to force ions or small molecules through which through membrane against their concentration gradient by using energy, by expending energy from ATP molecules. You know, ATP are the basic source of energy, right? Yes. So by using this ATP, by using energy, this transporters force ions or small molecules through membrane against their concentration gradient. So we'll see the details of this facilitated diffusion and active transport in the next pages.